Hi everyone, this is the 6th lecture for your chapter Cell. In the last class, we started with the cell membrane and we discussed the different models explaining how the cell membrane is formed, okay? And what are its components, what are the proteins, how the phospholipids are arranged. All of that we understood through this final model and most accepted model, which is fluid mosaic model. Okay, so this is the fluid mosaic model. We discussed that there are such phospholipids arranged in bilayer, so it is like a sea, and on the sea you have some icebergs or boats rather, which are floating, and some of them which are actually forming integral part. Okay, so they are integral proteins and some are just extrinsic or peripheral protein. Okay, and the membrane is fluid in it is a quasi-fluid kind of membrane, which means it is able to move around. As you can see in this diagram, there are a lot of folds. Okay, so it is very fluid. It is like let us say a flag flying in the air. Okay, how it moves, undulations they are called. So such kind of movements can be seen on this cell membrane. Okay, now we move ahead and we discussed why the plasma membrane structure is asymmetrical. Asymmetrical matlab it is not the same on the both side. Okay, normally what does symmetry mean? Symmetry means that if you take anything, let us say I'll draw some heart. Okay, now this heart, if I cut it from the middle, it should be symmetric, meaning on the left and on the right it looks the same. Then it is called symmetrical. Asymmetrical means that it does not look the same. Okay, so here, so here when you see, this was the reason I taught you different kinds of phospholipids. Okay, there are different phospholipids present in both layers. We will start with the outermost layer first. The outermost layer has phosphatidylcholine. Okay, then it has sphingomyelin. Sphingomyelin has sphingosine as the alcohol. Okay, and so sphingomyelin. Then the inner membrane. Okay, this is the inner membrane. So this inner membrane has phosphatidylserine, phosphatidylinositol, phosphatidylethanolamine. Okay, these three are very, very, very rarely found on the surface. Okay, and phosphatidylcholine and sphingomyelin are very rarely found on the inner side of the membrane. Okay, in fact, in some cancers, to detect the cancer, what you can see is their membranes have different phospholipids. So this just for an example, phosphatidylserine, if it is normally seen on the inner membrane, suddenly you will start seeing a lot of phosphatidylserine on the outer membrane as well. Okay, so this is how we can detect that there is something wrong with the cell. Clear? So there are different phospholipids present in both the layers. Amount of carbohydrate present in outer layer is higher. Okay, inner layer rarely has any carbohydrate. Some of them may be exception, but mostly glycolipids, which means carbohydrate plus lipid, and also what are not shown here are the glycoproteins. Okay, such branches you must have seen in different diagrams. Okay, so these are nothing but these are oligosaccharide moieties, which are carbohydrates. So carbohydrates are seen on the outer membrane but not on the inner side of the membrane. Okay, this is a bilayer, right? So we're talking about the outer layer and the inner layer and why there is asymmetry. Some proteins like spectrin are present only on inner membrane or inner surface of plasma membrane. Okay, spectrin, which has some protein like this called N-chirin, and with N-chirin there will be spectrin running through the whole cell membrane, okay, across the cell. Its function is to provide stability and structure to the animal cell. Okay, so it provides structure and stability to the animal cell, and it supports the plasma membrane. Okay, so example is spectrin, which we had discussed in the last class. Okay, go back and study. We had discussed spectrin in peripheral protein, right? So the peripheral protein found inside and the peripheral protein found outside are very different. So these are the reasons why there is asymmetry. Apart from this, you also see receptors, okay, protein receptors which are present on the outside, while the receptor, after it sends the signal forward, there will be different enzymes which are present on the inner membrane, okay, inner layer of the membrane. So these enzymes then they convert, let us say, ATP to something called cyclic AMP. Wait, let me draw again. Here you have the receptor. As soon as some hormone or some molecule, some signaling molecule, okay, it can be any hormone, it can be anything else. As soon as it attaches, the receptor changes its conformation. You must know that proteins are not stable, okay? They can keep moving around. They can have different, what you can say, structures, okay? They are bonds. So this will send signal to the inner side of the membrane where there are some enzymes present. For example, this enzyme is called adenylyl cyclase. What it does is it converts the ATP, here ATP, into cyclic AMP. Okay, cyclic AMP. Now this cyclic AMP is known as secondary messenger. Okay, and secondary messenger, this will go ahead and attach through some proteins and cause either DNA to form mRNA. Okay, so you will have mRNA. From mRNA you will have proteins, and these proteins will show their effects. Okay, whatever the signal has brought the message, that message will be transcripted, translated, and then finally shown. Okay, so such kind of function of receptor are present only on the outer membrane, while the or sometimes integral proteins, they are integral proteins, mostly they are outer membrane proteins, and some of these enzymatic proteins are inner membrane proteins. So this way also there is a difference between the outer layer of the cell membrane and the inner layer of the cell membrane. Clear? Now let us talk about some specializations of plasma membrane. Okay, some places, some organs where the plasma membrane has taken up some new or some special function. Okay, first example is of microvilli or brush border system that you see in digestive system. Okay, so in digestive system, this is your small intestine. When you cut your small intestine, you can see such finger-like projections which are called villi. When you take this villi, this is a single villi, okay, and you zoom this surface. Can you see the small box? Okay, when you zoom this surface, you see particular cells. Okay, so you can see each and every cell here, and on top of these cells, you see something called microvilli. Microvilli are nothing but they are just projections of the cell membrane. Okay, so for example, let me draw a cell. So if this is the cell, this is the normal cell. Okay, it does not have microvilli. And this is the cell which has microvilli. It is like small hair-like structures on the top. Fine, it is just a projection of the cell membrane. There will be a lot of cytoplasmic, cytoskeleton elements involved in making these kind of projections, these kind of microvilli, which we may study when we study cytoskeleton. Okay, fine. So brush border system, in small intestine you see such microvilli. Then in proximal convoluted tubules of your kidneys, okay, in PCT of the kidney, what I mean is nephron. Okay, in the kidney you have nephrons, which you draw something like this. There you have a nephron. Chalo, no problem. Fine. So this nephron you have, here there will be glomerulus, this is the Bowman's capsule, then you have neck, then you have proximal convoluted tubule, and then this will be the distal convoluted tubule, right? So in this proximal convoluted tubule, you find some microvilli-like structures, okay, which are just specialization of plasma membrane. One more thing I had to tell you was in this brush border system, okay, in the small intestine, these microvilli also have some enzymes, okay. On these microvilli, there will be presence of enzymes, and these enzymes will help in breakdown of disaccharide into monosaccharides, dipeptide into monopeptide, dinucleotide into mononucleotides, and so on and so forth. So the final breaking down of into small molecules will be done by the brush border enzymes, and the brush border is made up of nothing but specialized plasma membrane called microvilli. Then caveolae is seen in nephrons and neurons. Caveolae are nothing but cavities, okay? Like this, you will have such kind of a cavity on the cell membrane, and this can be actually used for multiple purposes, okay? These are not to be studied in detail for you, so it is okay if we skip it. Fine, just remember in case if they ask, cave means like a den, okay? So caveolae is actually some cave-like structure seen in the plasma membrane. Then you have sterocelia. Sterocelia means these are cilia, but what is special in them? So they are actually like this, okay? Let us see here itself in this diagram. So you have something called organ of Corti in ear, right? In your ear. So what happens is in that organ of Corti, there are such cells, and these cells have these sterocelia. Now there is fluid in this surrounding membrane, okay? What happens is now whenever there is sound wave coming, okay, sound wave, the fluid will also vibrate. When the fluid vibrates, the cilia, which are normally upright, they start moving on one side or they start moving on the other side. This message is then passed on to the cell and then to the neuron, and that is how you actually you are able to hear, okay, what somebody is saying. So sterocelia are present in organ of Corti. Some of these type of cilia are also present in epididymus of testis. Okay, if you remember the structure of testis, it was something like this, and then you have here epididymus. Okay, epididymus is nothing but it has a lot of such tubules through which the sperms have to pass, and only after they pass, only after they mature in these tubes, they are allowed to enter into the vas efferentia. Okay, so epididymus is this portion of the testis, and it also has some cilia-like structures. These cilia-like structures will help in moving the sperms forward, okay, because there are so many tubules, the sperms have to move ahead. So sometimes these cilia are also used to move the sperm ahead, or so in the epididymis in testis. Okay, then in prokaryotes we have already discussed, they have mesosomes. Mesosomes are nothing but they are some special structures of plasma membrane, and because the prokaryotes do not have organelles, so for compartmentalization, okay, or to make sure that not all reactions are occurring in the same cytoplasm, mesosomes are present. For example, if this is the bacterial cell, and there will be something like this, this is a mesosome. Okay, now at this mesosome, let us say there will be glycolysis happening here, then Tca cycle. Now mind you, they do not have mitochondria, but they do aerobic respiration, okay, the Tca cycle and everything, electron transport chain, so they will all be present here. Okay, this enzymes and everything will be collected here, the Tca cycle will be running close by, and so this is how they help. Okay, it creates a sort of cavity here, can you see, a sort of small space. Okay, so some particular reaction might be taking place there, some other reaction taking place somewhere else. So in case of prokaryotes, the specialization of plasma membrane are called mesosomes, and mesosomes help in such kind of role. Okay, they help in sort of compartmentalization of the cell. Okay, not too much, but they allow some specific reactions to take place in a particular side of the cell. Now we move on to the cell junctions. Cell junctions matlab, if you okay, here you can see there are a lot of cells. Here the uppermost dark pink layer is actually made up of cells. Okay, all of the layers are made from cells, but that dark pink layer is important. Okay, just notice that how all the cells are arranged one after the another. Okay, attached to one another. So how do they maintain this shape? Okay, let us say if I touch it, will these cells fall apart? Will they move out? No, right? They will stick together. So how are they sticking together? How are these cells forming something like some structure like this, like a villi? And how are such villi forming a structure like this? Okay, how is small intestine, let us say, maintaining its shape? So the cells actually have some junctions. Junctions meaning where two cells meet. Okay, and these junctions are important to answer all the questions I just asked you. Okay, chalo, without wasting time, let us start. Tight junctions. Tight junctions, from the name itself you can say that it will be very closely attached, right? Let me give you an example. Is there blood in your veins and in your arteries? Yes, right? And it is flowing always? Yes. So how is it not flowing out of the arteries? It is always flowing inside the arteries and veins, right? It is not leaking out. So it is not able to leak out because the membrane, okay, not the membrane, the what you can say, cells of the endodermis. Let me just draw. Chala. If this is your artery, then there will be cells here, right? There will be cells. Okay. Now these cells are not allowing the blood to pass out, okay, to leak out. So between these cells, which are arranged very close to each other, there are tight junctions. Okay, they keep the cells very tight, very close. So tight junctions are when the plasma membrane of two nearby cells are fused tightly. It is found in epitheliums and endotheliums. So the outer lining or the inner lining, where cells are supposed to be very closely attached, okay, as to not allow anything to go out or anything to come in. Okay, fine. Let us see here. You can see this is in case of, again, digestive system. You have mucus layer here, uppermost. Then you have the apical side, okay, which is closer to the mucus, is called apical side, and this is here will be the basement membrane. Cells are not just found randomly arranged. Okay, they have a membrane which is made up of different kinds of cytoskeleton elements or fibers and proteins. Fine. So on these basement membrane you have cells which are attached like this. So this is called basolateral surface. Okay, now basolateral surface, between the two cells you can see such kind of tight junctions where they are stitched together, you can say, almost. Okay, as you can see here. Fine. Here you see some proteins, and these proteins are helping to stitch the two cell membranes together. Okay, this will be the cell membrane of one cell, and this will be the cell membrane of another cell, and they are attached at certain points with help of proteins. These protein examples are given here: catenins, cadherins, occludins, actin, etc. There are many proteins. Fine. The next type of junctions, there are different junctions. Obviously, they have different functions. So desmosomes are button-like thickening between the two cells. Okay, they look like buttons. This is not a desmosome. I'll show you what a desmosome looks like. There are tonofibrils, which are nothing but keratin filaments. They are just called tonofibrils, attached to these thickenings. It provides rigidity to the epithelium, and it is found in epidermis. Okay, epidermis means your skin. It is found in skin. Epi means upper, and dermis means skin. Now let us see. Okay, these are the tight junctions between the two cells. These are the proteins causing. These are also called cell adhesion proteins. Okay. Then you have desmosomes, which are shown here. Here are the desmosomes. Okay. What you see is this part here. Now somebody will ask me, why not tight junctions everywhere? Why not tightly close every place on the cell membrane between the two cells? Imagine if your cell has taken up a lot of nutrition, a lot of water, it will need to expand. Where will it expand? It cannot expand if there is no space, right? If there are tight junctions. So in between there are some such cavities, some such spaces allowed where the cell can move around a little bit, or it can grow if it needs to. Fine. Okay. So at such places where there is some distance between the two cell membranes, you find desmosome. Okay. The yellow fibers you see are what are called tonofibrils, okay, which are nothing but keratin filaments or your cytoskeleton intermediate filaments. And these are the cell adhesion molecules, proteins rather. Fine. The last, second last type of cell junction is called gap junction. Gap junction, literally there is some gap, there is some pore which is present. Okay, if you see here, you can see a canal, right? Do you see a canal like that? So two cells are connected. Only in some cells you find such kind of a structure. Okay, when the two cells have to be connected for some signaling purposes. Okay, there you find gap junctions. Now these gap junctions, whatever the channels you are finding, whatever the channel proteins you are seeing, are actually called connexons. Okay, these are the connexons. They are made up of proteins, and in between what you see, that is the hole, that is the pore through which substances can be transported. Okay, signals can pass through. In gap junctions, two cells are connected to each other by protein tubules called connexons, which we just saw. Okay, its function is to transport small ions. Okay, small ions will just diffuse rather from one cell to another, and also some signaling molecules will be able to move from one cell to another. Final type of cell junction is plasmodesmata. Plasmodesmata we have already studied in cell wall. Okay, it is basically a pore, and through that pore you have smooth endoplasmic reticulum which are connected. Okay, from cell 1 to cell 2 there is this desmotubule through which two smooth endoplasmic reticulums are connected, right? So this is again connection between the two cells. It allows things to pass from one to another. It also helps in simplest pathway of water absorption, and transport of other molecules, signaling molecules also happen through this plasmodesmata. Now we move on to the transport across the membranes. Transport we will actually be studying in the chapter transport in plants. Okay, but here also we will discuss a few because they are directly related to the plasma membrane. Okay, so in plasma membrane there could be different types of transport. The first type is non-mediated or passive transport. Mediated matlab with help of something, and non-mediated means without help of anything. Okay, or passive means without use of energy. There is no use of energy. Example is diffusion. For diffusion, just imagine this is one membrane, and outside the membrane, let us say this is a bacterial membrane. Okay, there is a bacteria here. Okay, this is true for all the membranes. Okay, not only for bacteria, it is true for fungus, it is true for plants, it is true for animals. Fine, let us start. So you have put some glucose outside. Let us say there are 100 molecules of glucose outside, and there are only 20 molecules of glucose inside. So what will happen is, this is again just an example. What will happen is the molecules of glucose are higher outside and they are lower inside. So these glucose molecules will be randomly moving around. Okay, this is called random motion, and randomly they will also diffuse towards their lower concentration. Okay, how long will this happen? So till there are 60 molecules of glucose outside and 60 molecules of glucose inside. Okay, this is called equilibrium. Till equilibrium is reached, the molecules will diffuse. There is, again, I'm telling you, there is no use of energy. They are just moving around in random motion and they are diffusing, okay, from their higher concentration, from 100 molecules to their lower concentration, 20 molecules, till on the both sides, 100 plus 20 is 120, divided by 2 is 60. So on the both sides there are 60 molecules of glucose. Then the movement, that exchange will stop. Okay, so this is diffusion. Fine. Then there is osmosis. Osmosis is just diffusion of solvent molecule. Now what happens is, in osmosis you use a semi-permeable membrane. Okay, semi-permeable membrane. Semi-permeable meaning it will allow some things to pass but not allow all things to pass. So what will it allow to pass? It will allow water molecules, H2O, which is a solvent. It will allow solvent to pass, but it will not allow solute to pass. Solute will not be allowed to go in. Okay, so what will happen is, let us say we'll take the same example again, but now with a semi-permeable membrane. Okay, this is the semi-permeable membrane. You have 100 molecules of glucose outside, you have 20 molecules of glucose inside. But because semi-permeable membrane will not allow the solute to pass, so these 100 glucose molecules cannot really diffuse. What can diffuse? So water can diffuse. Okay, so this water from the cell will go out. How much of it will go out? Till the volume of this side increases. Okay, the volume keeps on increasing. How much will it increase? Till the time that there will be 20 molecules on this side. Okay, let us say 20 molar and 20 molar on this side. It will get diluted. Okay, it gets so much diluted that there will only be 20 molecules on this side as well. Clear? Obviously there will be 100 in total, but the volume will increase so much that in the vicinity, in the nearby region, in this much region if you talk about, you will only find 20 molecules of glucose. The others will be very much diluted. Okay, so this process of movement of solvent particles, okay, solvent molecules, it could be water, it could be any solvent, from its higher concentration. Okay, here because there were just 20 molecules of glucose, the concentration of water was higher. Okay, the solute concentration is low but water concentration is high. On the other side, solute concentration is high but the water concentration is low. So movement of solvent, okay, which is H2O, from its higher concentration to its lower concentration is called osmosis. Okay, osmosis can be endoosmosis or exoosmosis. Let us discuss with help of this diagram. We have rather used this RBC. Okay, now if you put RBC in hypotonic solution, hypo means less and tonic means it has less amount of solute. Okay, it is hypotonic. So in hypotonic solution, what will happen is the concentration of water outside is higher and the concentration of water inside is lower. So water will move from outside into the cell, okay, into the RBC. So this process is called endoosmosis. Okay, endo meaning inside and osmosis, water moving inside the cell. Then if the concentration of water outside and inside the cell are same, then there will be no net exchange of water. So this is called isotonic condition. Iso means same. And then finally you have hypertonic solution. Hypertonic meaning the concentration of water will be higher inside. Okay, the concentration of solute will be higher outside. So the water from the cell will move out. And this kind of movement is called exoosmosis. Fine, all of these are non-mediated or passive transport. Fine, there is no energy used in the process. Then you have mediated transport, which means you need some help, normally from a protein. Example is facilitated diffusion. Facilitated diffusion is just diffusion, but you require a protein for it to diffuse. Okay. Again, let us take example of that glucose. You have 100 molecules of glucose, you have 20 molecules of glucose inside. But because glucose are polar molecules, okay, they are polar. So polar items or polar molecules cannot cross the cell membrane. They cannot, because the cell membrane is not polar, in between. So you require some protein, some channel protein through which these polar molecules can move in. Okay, and these proteins are channel proteins or carrier proteins. They do not take any energy. Okay, they do not require any energy, but they will be helping in diffusion of the molecules according to their concentration gradient. Then you have active transport, in which you require energy for moving things, from moving the molecules from one side to the other. And you can see, because you are using energy, you can even try to go against the concentration gradient, even though the concentration outside is low and inside is high. You will still be able to grab each and every molecule and pull it inside the cell with help of energy, of course. Okay, so that is active transport. Active meaning you require energy. Fine. Then there can be co-transport. Co-transport is you move two molecules together. Okay, like here A and B. Only if both of them are present you can help in moving them inside. I'll give you an example of ORS. Have you heard of ORS? ORS, oral rehydration system. ORS is actually nothing but sugar plus salt plus water. Okay, why is this combination given? So in your intestine you have such kind of cell membrane where glucose cannot be carried inside alone. Okay, so glucose, let us say here B is glucose. This glucose will not be able to go alone inside. It requires another molecule, okay, something else. What does it require? It requires sodium. Only if glucose and sodium are both present together, they will be taken up and they will be able to enter the cell. Okay, so this is an example of symport. And then you have antiport. Antiport is one molecule is taken from outside to inside and the other molecule is taken from inside to outside. The easiest example is the sodium potassium ATP is pumped in your neurons. Okay, they will take two potassium from outside to inside and 3 sodium from inside to outside. Okay, antiport is also called counter transport. They are one and the same. Finally, you have vesicle mediated transport. Okay, this is something new for you, I think. In vesicle mediated transport, you have endocytosis, which can be pinocytosis or phagocytosis, and exocytosis. Let us see it here. Phagocytosis. Whenever you hear this term, at least whenever I hear this term, I think of amoeba. right? In your lower standards you have studied phagocytosis. It is nothing but when there is a solid particle, a food particle, it could be bacteria, it could be just some food particle, it could be anything, okay? It could be an algae. What happens is your plasma membrane starts to form something called pseudopodia. Pseudo means false, okay? False, quota, false, and podium means legs, because it looks somewhat like legs, okay? So it starts forming such pseudopodia, and these pseudopodia will keep on developing till they merge, till they meet each other. Once that happens, this food particle which was outside will now be inside this vesicle, okay? So this here is called a vesicle, fine? This here, once the pseudopodia meet and they form this kind of spherical structure, that is called a vesicle, and this vesicle will move inside and form a phagosome, fine? So this is phagocytosis. Then there is phagocytosis, there is some solid particle. In pinocytosis or pinocytosis, you take up extracellular fluid. For example, if there is glucose outside or if there is sodium outside, then these are not solid particles, they are dissolved in water. So what happens is the cell just takes up this fluid, it forms this sort of vesicle, okay? And this is how the fluid drinking, or it is cell drinking, okay? Pinocytosis is taking up fluid, phagocytosis is taking up solid particles. And finally, there is receptor mediated endocytosis. There are receptors on the cell membrane, as we had discussed. These receptors are very specific. They will allow only one thing to attach, okay? They are very specific for any one thing. For example, you can see here there are square type of molecules and star type of molecules. But these receptors can bind only with the star type of molecules, okay? So as soon as this star type of molecule binds, these what you can say, pit, okay, there will be a slight curvature, so it will start forming inwards, okay? It will start becoming like this, and then finally it will fuse like this, and once it fuses, then this vesicle will become like this, okay? So you have receptors here, you have the molecule here, and they are all present inside the vesicle. So this is called receptor mediated, okay? With help of receptor you are doing endocytosis. Endocytosis is taking something inside the cell. The exact opposite reaction, okay? If this vesicle goes and fuses, and the whatever was present inside, it could be waste, it could be proteins, everything is thrown outside the cell, then this is called exocytosis, fine? So these are the mediated transport, and in mediated transport, vesicle mediated transport, okay? You have normal mediated and vesicle mediated transports. Now we move on to the last part, just a summary, quick summary. Functions of the plasma membrane: it causes compartmentalization, okay? It will separate the outside environment from the inside environment of the cell. Then functions of the plasma membrane: compartmentalization we discussed, then protects the cell wall, protects the cell from injury. It will not allow the cell to become injured, okay? As much as possible it will try to bend itself rather than injure the cell, okay? That fluid nature comes in handy here. Then it allows the flow of material and information within the cell and between the cells. Plasma membranes of adjacent cells form various types of junctions. We saw 4 types of junctions: tight junction, desmosomes, then we saw gap junction and plasmodesmata. It is selectively permeable, that is, it will allow only selected substances to pass inwards. Okay, the membrane are impermeable to the other molecules. Different types of transports are used, which we just discussed. It possesses recognition protein and points of attachment. I'll tell you what this means. Now, ever wondered like how your kidney cells always recognize each other, or they are with other kidney cells, okay? Or cells of your liver will be attached to the other cell of liver, fine? So what happens is, if you take, let us say, some liver cells or hepatocytes and some cells from your kidneys and mix them together, okay, grind them together, so that they do not break, okay, you do not have to break the cells, you just have to separate the cells. Now you have a mixture of kidney cells and liver cells all mixed up. If you allow it to stand, okay, you allow it to stay like that, and then after, let us say, 1 day, okay, after a day you come back and you see, then you will find that the kidney cells would have come together and formed a group, and the liver cells would have come together and formed a group. This is because they have recognition proteins present on their cell membrane. So one liver cell recognizes other liver cell and tries to get attached to it. Again, this is how the cells try to regroup. Fine. Glycoproteins on the cell membrane determine antigen specificity. Example is of blood group antigens on RBC. Like there could be glycoprotein A, then your blood group will be A. Example. Then B type of protein will make your blood B. If you have both type of glycoproteins, then your blood group will be A, B. So these are called antigens. Okay, antigen. Clear? They are also present on the cell membrane. Cell membrane has receptors for certain hormones and takes part in signaling. This we can discuss when we come to the hormones part, okay, chemical coordination part. You will study a little bit of signaling if I am the one who will be teaching you, okay. Then plasma membrane contains enzymes for performing certain reactions on their surface, like ATPases. Then I had told you about adenylyl cyclase, which converts ATP into cyclic AMP. And that cyclic AMP is then a signaling molecule. It will cause some or the other reaction. Mostly it causes the DNA to transcribe and the mRNA to translate and form new proteins. Okay. Cell membrane of prokaryotes possess the electron transport system. Because prokaryotes do not have separate mitochondria and separate chloroplast, their cell membrane is the place where this electron transport systems are active. Membrane enfolds are used for bulk intake of material by endocytosis. Okay, endocytosis, we saw the 2 types, phagocytosis and pinocytosis. And as microvilli, the membrane becomes specialized for absorption of substances. Okay, microvilli increases the surface area, and as the surface area increases, more and more substances can be absorbed. Secretory, excretory, and waste products are thrown out through exocytosis. Okay, secretory matlab something that the cell is secreting. It could be a hormone, it could be some protein, that is secretion. Excretion is removal of waste products, okay, like ammonia, urea, uric acid, all of that is excretory and waste products. So all of that is thrown out through exocytosis. In nerve cells, okay, in neurons, you have nerve impulse transmission or action potential. So this action potential or nerve impulse transmission happens or takes place on the plasma membrane. Okay, so it is important there as well. Then plasma lemma, which is another name for plasma membrane, provides sheath for cilia and flagella. Now this we will study a little bit when we study cilia and flagella. But in case of eukaryotes, the cilia and flagella are covered by a sheet of plasma membrane. Okay, so this is called plasma lemma. It helps in movement of some cells by pseudopodia, example amoeba, which we have already discussed. So here we end the cell membrane. Okay, see you next time.